This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1207, recorded on April 4th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And it's a beautiful early spring day here in Western Mass at 65 Fahrenheit, 18 C. Got a few clouds in the sky, and it's a little breezy, but generally wonderful weather.
21 C and cloudy here in Chelsea. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We've got 85 degrees and, well, kind of high overcast. A diffuse Dixon is over in the corner there, over my left shoulder.
Diffuse Dixon.
Yeah, and they're talking about rain, but it'll show up tonight, I suppose. We can use all the rain we can get. There was a bunch of rain while I was gone last week that I missed. That's good.
Yeah, it rained a lot this week here. Cloudy. It's okay. It's warming up. For once, I don't feel cold. I'm very happy about that. If you enjoy these science programs, we'd love to have your support. Go to microbe.tv/contribute. We don't do ads on our programs, so there's the only way to raise money to pay for them is by your donations. We exist on your generosity. And I know in the U.S. there are still a lot of people who are generous, so we depend on that.
Fortunately, we do not receive any federal support.
Yes, we don't. Or else,
I mean, in the past, I would have said we'd love to have federal support, but no, not these days.
You know, I always tried. I did try to get NIH support years ago, but there was no mechanism.
Right.
So that's that. And that's fine. Now we don't have to worry about them taking it away, right? This trio, Rich, Allen, and me, is reminiscent of the early days of TWiV, when Dixon used to take off for extended periods. So if you look in the low-numbered TWiVs, you'll see a lot of this. ASV, American Society for Virology, the 2025 meeting, hosted by McGill University, will be held in Montreal at the Palais des Congrès, July 14th through the 17th. You can go to the website and check it out, asv.org/ASV2025. There's the program there, and you can also see the satellites and the plenary sessions. Deadlines. Deadline for ASV Cares and Kitty Corps, May 9th. It's about a month away. These are dependent care benefits for ASV members. And if you have your concurrent workshop assignments, you can plan what on-site child care hours to register for. Early bird registration, where you save money, ends May 12th. Only one bit of news this week. I thought you had enough bad news last week, which is not to say there isn't more bad news this week, more firings, more cuts. Princeton University and Brown are now being threatened with having money withdrawn from them. This is the death of Klaus Conzelmann. I got a letter from Stefan Becker this week, March 25th. He will always be remembered in the NSV community. The NSV means negative strand virus community, as the guy who first successfully established a reverse genetics system for a negative strand RNA virus, for which he received the Loeffler-Frosch Award of the German Society for Virology in 1995. The key was he had to make plus strand RNA and put it in cells, not minus strand. Okay.
Yeah, which virus was that?
He did it with, maybe it was rabies, because they say his pet virus was rabies.
Right, rabies or VSV, maybe.
Either that or VSV. Over the past decades, he has contributed to our understanding of innate immune sensing of RNA and viral immune evasion. He also developed rabies vectors for neurotracing, used to map neuronal circuits. He was professor for experimental virology at the Max von Pettenkofer Institute and the Gene Center of the Ludwig Maximilians University in Munich. Retired 2 years ago, and he has trained and guided numerous mentees who are now in leadership positions. To me, Klaus was a wonderful mentor and friend. He introduced me to virology and helped me become the scientist I am today. I see many of his traits in myself, his curiosity and eagerness, his encouraging and supporting mentorship style, his Southern German sense of humor. Stefan. Okay, I want to know what a Southern German sense of humor.
I have no idea.
Tell me a joke in a Southern German sense of humor.
No idea. Somebody, we need some comeback on this.
I'm sure there are people listening who can.
I wonder if you can even Google Southern German
probably
sense
could.
of humor and come up with something.
Well, it must be different from the Northern German.
Must be.
I suppose.
Yeah.
Right? The Northerns and the Southerns didn't always get along,
No.
right? Down south they're the Bavarians, and up north they're the Prussians. And Bavaria is very technologically advanced. They make a lot of stuff.
Here's a
BMW.
here's a Reddit post that says very similar to British, very dry, sarcastic,
Ah.
deadpan.
I see.
I don't know if that's German generally or Southern German.
Yeah,
But
it's not
there you
very
go.
interesting. It's not a very interesting sense of humor.
I don't know. I'm
Depends.
kind of into dry deadpan.
Yeah,
sense.
no, I like that.
You know.
I like that.
I know. I liked. Who was the guy? Not. I can't even remember any of their names. The guy he had a ponytail and a beard. He was kind
I'm
of. a
thinking
hippie
you're
type.
thinking George Carlin.
George Carlin.
George
Yeah, when I'm
Carlin.
thinking,
Oh, yes.
what humor would Vincent like?
Yes.
It's George Carlin.
George
Yeah,
Carlin.
Cynical.
George Carlin.
He was
Yes,
cynical.
very cynical.
Yes,
very cynical.
I do like George Carlin. 7 words you can't say on television.
Yeah. And he's got a whole rap on stuff.
On stuff, yes.
I think
Right?
we've picked his thing, but maybe it was you, Rich, who picked him.
I
I
George
think
Carlin.
I may
that
have
sounds like
picked-
Dixon.
I may
Oh,
have
maybe-
picked one of
Yeah,
his monologues.
maybe you.
So, what kind of humor would he be classified as?
I'd call it cynical.
Just cynical.
Yes,
very
he's not
cynical.
He's not typically American.
I think of that as pretty typically America. It's a strain of American humor.
Yeah. Also, I also liked Rodney Dangerfield.
Oh,
More of a classic comedian.
I never
Yeah.
really respected him.
Yeah, Dangerfield is sort of verbal slapstick.
Yes.
You know,
Exactly right. I liked that. I think to a certain extent that was entertaining. But George Carlin, really good.
Yeah.
Really liked him. There's another guy I liked who I can't think of.
did we mention this TWiV episode with Klaus on it?
Oh, we didn't. Klaus was on TWiV 102. Oh my gosh, that's so long ago.
I know. It was a
Never
big
gonna-
deal to celebrate TWiV 100.
100.
This was with me, Andy Baker, Klaus Conzelmann, Peter Palese, and Katharina Eisenacher.
2010, man.
Yeah, I mean, I had such a thing to set that. If you look at the picture, you can see all the wires, and I have a recorder right there. Oh my God, it was really difficult, but we pulled it off. It's a lot of fun. So, Catch Me If You Can. It was the name of the meeting, I think. So anyway, TWiV 102, October 10th, 10-10-10. Whoa. Cool. All right, two papers today for you, a snippet into paper. Now, the snippet, you're going to summarize it for us, Alan?
Yeah, I can actually do. So I was going to write up a little summary of this, but then I looked at the author summary and I said, I can just read the author's summary because this is a pretty good.
Yeah, it's fine. I just
good
want to
quick
mention
rundown.
why I picked this. Everyone's worried about avian influenza in the next pandemic, but I think pigs are a likely source of the next pandemic virus, and this will show you some reasons why.
Yeah. Yeah. So I'm just going to, I'll rephrase a couple of things in the author's summary. So they're looking at swine flu from the pandemic 09 strain, if people remember that. So they say the diversity and evolution of influenza A virus in pigs is linked to the emergence of flu with zoonotic potential. Human to swine transmission of the 2009 H1N1 pandemic, which I'm just going to call Pandemic 09, lineage repeatedly occurred across the past decade and has increased genetic diversity of the virus in pigs. Sporadic swine to human cases are associated with those viruses. So they measured the frequency of human to swine transmission of the pandemic lineage from 2009 to 2021 and determined how this affected the diversity of flu in swine and predicted zoonotic risk. They found 370 separate human to swine spillovers. The frequency of interspecies transmission increased when the burden of flu was highest in the human population. Makes sense. Most of these spillovers were single events without sustained transmission, but a small subset resulted in emergence, persistence, and co-circulation of different Pandemic 09 genetic clades in U.S. pigs. Each of the Pandemic 09 represented, coming from different spillovers, was genetically and antigenically different from the human seasonal vaccine strains. So the persistence of Pandemic 09 within pigs resulted in at least 5 recent swine to human transmission events, suggesting that controlling flu infection in humans working with swine could minimize spillover into pigs, reduce resulting genetic diversity of flu in pigs, and reduce the potential for swine to human transmission of flu that would have zoonotic potential. So the importance here is you see a lot of flu going from the humans into the pigs. Some of it comes back into the humans, and a lot of the time it is antigenically different because it changed in the pigs and would potentially evade vaccination.
You know, I used to teach this stuff to medical students many eons ago. And sort of the dogma there was that the reservoir for influenza was birds. And I think that's still a reasonable dogma. But in terms of, and, you know, we knew that pigs were a mixing bowl, okay? But the dogma was that most of the pandemic stuff in humans came ultimately from birds, or at least that was maybe I was teaching it wrong, but that's the dogma I taught.
That was pretty much it, yeah.
Okay, but this is sort of a facepalm for me, because we've known that pigs played an important role for a while, and I've kind of ignored it because I'm imbued with the dogma. But this really makes the point. Pigs are important.
Well, and I think part of the dogma has been that birds are where, you know, that's kind of the ancestral host for all these flu viruses that are circulating around in the wild. But pigs are a very good intermediate host, and that's why we've had 2 swine flu scares, including the 09 pandemic.
Yep. All right, so this is a PLOS Pathogens paper.
Open access.
Open access, reverse-zoonoses of 2009 H1N1 pandemic influenza A viruses and evolution in U.S. swine results in viruses with zoonotic potential. This is all about American pigs, folks.
Yep.
Yeah.
American pigs.
And it's a good old American pork.
American pork.
The first author is Alexei Markin. Senior author is Tavis Anderson. And the team is at the U.S. Department of Agriculture, Agricultural Research Service in Ames, Iowa, and Iowa State University in Ames. So this is an all Ames team.
I hope they're able to continue their work. That's
I
all
hope so.
Yes, I hope so too. I was about to note that this is federally funded.
Yep.
Most of the research we talk about is federally funded by either the U.S. or other countries.
So, as you heard, there's often human to swine transmission of influenza A viruses, and you may wonder why. Well, we have a lot of pig farms, and people need to take care of them, so they're pretty close to them.
Yeah.
Maybe walking next to them, right? Check them out and all that. And so they can give them viruses, and pigs can give them back to us, as you'll see. Currently, there are over 20 genetic clades of HA genes, H1, H1N1, H1N2, and H3N2 circulating in U.S. swine. And some or all of the 8 virus genes in these viruses are derived from an ancestral human influenza A viruses.
So this interests me. This is 20 H1 clades among the H1 to H3 viruses.
Yeah.
Because, we think of H1 is different than H2 is different than H3, and there are sort of definitions of what distinguishes one from another. But each of these gets subdivided so that within H1 there are a bunch of different clades. I don't know what defines a clade.
Yeah, a certain percentage of nucleotide difference.
Okay.
I don't know what it is offhand.
So
But what's
not enough to
interesting-
make it a different hemagglutinin subtype,
No,
but enough
no,
to
no.
make it a different clade within H1 or
Exactly
whatever.
right.
Okay.
And
Notice,
probably enough to evade immunity from the other.
That's what I was thinking.
yeah. Notice it's circulating in pigs currently. H1N1, H1N2, and H3N2. There's no H2N2. Not circulating anywhere, as far as I know. Ron Fouché told me that a number of years ago. That's why. So most humans, if you're born, let's see. So H2 arose in 57 and circulated to 68. So if you're born after 68, you have no immunity to H2. And so I don't, well, yes, I do, because I was living during the H2 pandemic. But anyway, the further we get away from 68, the more people are lacking immunity to H2. And Ron Fouché thinks,
I don't have immunity to H2.
yeah. So Ron Fouché thinks that's going to be in the next pandemic. It's actually in birds, but it's not in pigs.
So I
Anyway.
wonder what's the story with H2N2? Is there some sort of weird incompatibility there?
In pigs? I don't know. It's a good question.
H2N2 does exist somewhere, right?
It's in birds, yeah.
Okay.
It's in freezers and labs. As far as we know, it's not circulating in any people.
Okay.
Anyway, when these viruses are in pigs, as Alan said, they drift antigenically, and they may have no immunity to human immunity may not do anything to them. And so they represent a zoonotic threat. Now, this story, maybe most people don't know this, but the 2009 H1N1 pandemic virus, okay? In 1918, before 1918, there was no, as far as we know, no influenza virus in pigs. In 1918, the H1N1 that caused the big pandemic at the same time went into pigs and evolved in pigs for a long time, I say over 90 years here, and reemerged basically in 2009 to cause that pandemic. But in the meantime, it had drifted in the pigs, although not as much as it would drift in humans, because pigs don't live as long as humans, right?
And that virus had not only some of the original human sequences, but it had sequences also that it had picked up from both pigs and birds, right?
From two different kinds of pigs. Classical swine influenza, which is the 1918 lineage, and also Eurasian pig influenza virus, which is another thing, from a human virus, and also from bird avian influenza viruses. And so the 2009 virus got segments from all different kinds of animal viruses, and they estimate that the virus had been in pigs for 5 to 10 years prior to emerging.
So there may not have been flu in pigs before 1918, or there may have been. But after 1918, there definitely was. And since then, there have been other flu viruses that we know have been introduced and through modern agriculture repeatedly introduced into pigs.
Yes, and we'll see some of those today.
Two things come to mind. Modern agriculture is an important consideration in this because over this period of time, pig farming has undergone dramatic change.
Changed a lot.
And, that in particular, the way it's done now, I can only imagine, would contribute significantly to this. The other thing that occurs to me, and I'm jumping the gun here a little bit, but the magnitude of what's going on here with all of these spillovers and zoonoses and all of these evolution events, and yet the just few very significant zoonoses that come out of it really, to me, amplifies the issue of how difficult it is, statistically, if you like, or evolutionarily, to come up with a virus that's going to really trash humans. There's a lot of stuff going on here, but not many that come out and trash humans. And I'm thinking about, the notion that humans would create a virus deliberately to cause a problem. No, it's not happening.
We don't even begin to know how to do that.
No.
That's the thing. People think it's easy to do this. We have no clue.
No. Even the pigs don't know how. They're just doing, but they're doing experiments on massive scales.
Yeah, and that's the thing is if you keep rolling the dice enough times, they will eventually come up snake eyes 5 times in a row, and then you've got
That's why people who don't have any familiarity with virology, they just think, oh, you could just make it.
No.
You can't.
No.
You don't know what to do.
Yeah.
And then they make up stupid excuses, that's why the lab origin has zero credibility, because nobody knew. First of all, nobody had the precursor, but no one knew what to do with it.
So the pigs here are the virological equivalent of the monkeys and the typewriters producing Shakespeare.
Yes,
that's right. Now in this paper, they want to look at this 2009 H1N1 pandemic virus, its entry. So it went into humans, and then of course it's gone into pigs multiple times since then, and then it's gone back into people. And they do a lot of sequence analysis, so they have many sequences of this H1N1, specifically the HA, the H1HA. Between 2009 when it emerged in 2021. So this paper is a couple of years old. It's 13,935 human and swine H1 sequences. And you can do all kinds of analyses with these sequences. You could look at the relatedness between viruses. You could look at timing of emergence. And they estimate that these 2009 viruses were introduced from humans to pigs 370 times in the U.S. following the pandemic. And then in the other way, there were swine to human transmissions 17 times since 2010.
So again, the humans are slopping more virus into the pigs than the pigs are getting back into the humans.
Yeah, and I wonder about this. Is that because there are a gazillion more pigs than there are people involved in this experiment? Is it just a target availability analysis?
Well, the people who are targets are limited, right? It's the people who work on pig farms.
Yeah.
So there's 20 times as many infections of pigs as there are infections of humans. There's got to be at least 20 pigs per human, maybe.
Sure.
And so maybe it's just a target issue.
They do note that of these 17 pig to human transmissions, 4 were in children. So I don't know. Sometimes they bring pigs to
I'm thinking the
fairs,
petting zoos, but also potentially, bring your child to work day, or
bring your child to work, or you bring virus home. Right?
Yeah.
8 were in adults. Some of these viruses some of the clades that disappeared in humans continued to circulate in pigs. So they're gone in humans, but they're still circulating in pigs. And of the H1 introduced into swine from humans, 45% were maintained longer than a year. I don't know how long a pig lasts. Is it about a year?
I don't know how that's. I think it's not much longer than a year these days.
Yeah. You get to a certain weight and that's it, right?
Yeah.
Yep.
Yeah, they raise them as fast as they can.
Now, not unexpectedly, when in years where there was higher human H1, there was more detection of H1 in pigs. All right, now where do these happen? The states with the most human to swine spillovers were Illinois, Iowa, Minnesota, North Carolina, and Indiana. Those are the most. And then they had other states, Missouri, Ohio, Nebraska, and Oklahoma. These are states with pigs, right?
Yeah. This is where the pig farms are.
Yeah.
So, I don't think we have any in New York and New Jersey, right?
I'm sure there are some
small ones.
Yeah.
Of these viruses in pigs, where they have the whole genome sequenced, over half of them maintained the internal RNA segments of the original H1 strain, but 49% of them had reassorted genomes, picked up segments from other viruses infecting pigs, of course. So basically, most of these virus events reflect transmissions from humans and transient transmission in pigs, right? It's not forever. You never know.
I wonder if some of that, the transient transmission in pigs, I haven't thought this through, has to do with the fact that the pigs don't last for longer than a year or so.
Yeah.
Yeah, probably. Yeah. Yeah, if you had a population that lasted longer, you would probably see more.
And I just looked up, and apparently 4 months to 1 year is a typical slaughter time frame for pigs that are going to be bacon or pork.
Okay, so they can also use this data to tell you how many HA genes, again, we're talking about the 2009 H1N1, are detected in swine in any given year. Okay? And always the number of human to swine was an order of magnitude higher than the number of swine to human spillovers. So we're giving them viruses more than they're giving us viruses, but they don't have pandemics because they don't live long enough, right? They looked at temporal relationship between the human and the swine detections by looking at the sequence. And for 86% of the swine HA sequences, the closest human was collected just before the swine sequence was collected, further supporting this idea that the primary direction is human to swine.
So we're the reservoir host for the swine.
Yes, we are. Certainly. Now, there are other viruses circulating in pigs, which are not the subject of this paper. This is just all about the 2009 H1N1. But there are unusual variants of influenza viruses that are circulating in pigs, and periodically they cause infections in people as well. So others are keeping an eye on that. Okay, now they say the one season that didn't follow this correlation between human and swine detections, in other words, more human, more swine, was 2020-2021.
Why is this year different from all other years?
You may remember that was the onset of COVID, where there was very little human influenza activity in the U.S.
Because there was very little human activity in the U.S.
So there were 18 confirmed H1N1 cases in 2020-21. Isn't that amazing?
Yeah.
So that's all you need to do to stop influenza. Stay home.
That isolation and masks and those measures that we took that actually prevented virus transmission.
Now, the thing is, you would think, so there are not going to be a lot of detections in pigs. They were pretty high in 2020, 2021. For pigs, it was life as usual, right? It exceeded the 2018, 2019 season. They had 183 detections in 2020, 2021. So they think that it's transmitting among the pigs from the previous years. And so there were fewer spillovers from human to pigs in 2020, 2021. But if you look at the sequences of what's in pig in that year, it's similar to viruses from earlier years spillover, 2019, 2020, and earlier. So, half of the viruses came from 2019, half, and other years, 2018, 17, 15, contributed different amounts.
So it seems like the virus is doing better in the pig host in a year where there are not a bunch more introductions from the human host.
Yeah, it's interesting. So maybe the new introductions compete out the others.
I was thinking either that or the constant spillovers, slopping more virus to the hogs, is immunizing them.
Could be, yeah. What they do note here, which is very interesting, is that they find evidence for transmission of some of these human viruses in pigs for more than 10 years. So it's not the norm, but some of them stay in pigs for over 10 years. So despite the age and all that we just talked about, it can go longer.
Well, and there are bits of the 1918 flu that hung out in pigs for 90 years.
90 years.
For sure. Mm-hmm. Yes. So, yeah, the 1918 variants over the years just stayed in pigs all those years. Yeah. Quite interesting. Okay.
By the way, I've just been looking this up. Flu does make pigs sick. Mm-hmm. I don't know that necessarily all of these things make all the pigs sick, or what's circulating based on illness all the time, but it does cause illness, and it's a problem for pig farming, and there are flu vaccines that are used in appropriate circumstances in pigs. I don't know that everybody or every pig is routinely vaccinated, but there are certainly circumstances where the vaccines are applied.
It's a problem because they lose weight, and that's not good when they get flu. And pigs raised for meat, you don't want them.
You don't want them losing weight.
Yeah. So 10 years. I'm really impressed by that. Some of these viruses, at least 10 years, but the 1918, even longer. Okay. Now, what about the other way? They looked into swine to human transmission, and they found 18 cases of swine to human between September 2020 and October 2021. That's the pandemic year. So pig farmers or the people who work on pig farms had to go to work, right? You can't just stay home because the pigs need your attention. So they went to work and they got flu from the pigs. I guess if they had stayed home, that wouldn't have happened.
And then we wouldn't have had meat.
Yeah, right. So one virus, Iowa 2021, was a CDC-confirmed swine origin infection in humans with a reassorted 2009 virus. And then they had 4 other strains, and their sequencing shows that they came from the pigs. They're definitely pig origin, so they're zoonotic origin. Now, they did some sequence gazing at these human isolates, right? And 2 of them had a amino acid change in the HA, which is glycine 155 ASP, which has been associated with binding to alpha-2-6-sialic acids. And they also had asparagine to aspartic acid at 129, which is associated with antigenic escape. So those were present in quite a few of the sequences.
So
basically, they probably arose in pigs and then, they transmitted into humans.
Those are adaptations that would provide for fitness in humans. Is that the idea?
I think they were providing fitness in pigs.
Ah, okay.
Right. They provide fitness in pigs, but they might help evade immunity in humans who've been vaccinated or exposed to the circulating strains.
They could, yeah. All right, so the last experiment, they have these isolates, these human isolates that came from pigs, right? And they want to know how human sera, or ferret sera actually made against the H1N1 vaccine strains from various years, 2009, 15, 18, 19, how they react with these swine strains. And these viruses or these HAs have been circulating in pigs for 1 to 7 years following the spillovers. Okay. So basically, all of them have at least a 4-fold drop in cross-reactivity with the ferret antisera. So basically, they've lost some ability to be neutralized by antibodies generated by the vaccines that are used in humans. So it's a part of the concern here, right? They're antigenically drifting, and so These are viruses that are in pigs, by the way. I misspoke. They're not human isolates, but they're pig isolates. And it's looking how they neutralize. And they have reduced neutralization with sera against the vaccine strains.
So that, that all fits the dogma that I used to teach in birds, only this is in pigs, which is that the viruses can hide in this other host and drift over time and come up with something that would infect humans, but to which we would have no existing immunity.
Exactly.
Boom.
Yeah. So that's the paper, and I wanted to raise awareness of these viruses in pigs because everyone's focusing on avian H5N1, but this is also important, right?
Yeah.
So,
and they point out in the paper, and this paper is actually came out in 2023, I think.
Yeah.
Before bird flu was as big a story, but they do point out that with bird flu circulating and now increasing, you've got more opportunities for that to get into pigs. And if you're introducing Pandemic 09 into pigs from humans all the time, and you've got this existing population, you're introducing another bird virus in there. It's a lot in the mix.
And there's a lot of bird virus in cows now, right?
Yes.
So
that's a good question, right? What's the situation in pigs? If you remember last year, there was some backyard pigs in Oregon, I think, that tested positive. And they've done some experiments where they infect pigs with the cow-derived H5N1. They get infected, they don't get very sick, and they don't transmit it. So, and there haven't been any other reports of pigs being infected in the U.S., although, maybe there are and we just don't know. We're not looking.
Might be a screening problem.
Yeah, but the risk appears at now relatively low. But here in this one, 2009 H1N1 frequently crosses back and forth between humans and pigs and humans. Many of them are maintained for more than a year. Many of them reassort with influenza viruses in pigs, and they drift antigenically away so that so they're away from humans and they're drifting. And if they ever got back into humans, we'd have to use a matched vaccine, I think, because the ones we're using aren't going to work.
370 spillovers from humans into pigs over a 12-year period is
that we know
quite
of.
Something that we know of.
In this sample.
Yeah, it's as we know of, because we're not looking at every pig on every farm, right? So there could be even more. But the pigs, so basically they can be a reservoir, like we've thought them to be a reservoir before. And, older clades can go from humans into pigs and then many years, and then pigs back into humans, like what happened in 1918 to 2009, essentially. So we have to, they say we have to keep doing surveillance, right?
Yeah.
We have to do more than we're having. Continued genomic surveillance paired with characterization of the viruses by antigenic studies, as we've seen here, and also to see if any H5 genes are getting in there as well. So it should be part of pandemic planning, the assessment of these swine influenza A viruses. In any other year, I would say it would be done. I'm just scared it's not going to be done because some idiot doesn't think it's important.
This is exactly what's getting cut.
And should not be based on what one person thinks.
No.
It should be based on a committee. That's how science works, right? But we're in very uncharted waters, folks. So if you're worried, you should be.
Yep.
But I think most Americans are not worried.
I think people are getting a little more worried now that everything's going to cost so much more.
Yeah, money.
I think this is getting people's attention.
Follow the money.
Yeah.
It's another story entirely. Yeah,
Yes.
money. But cutting all the science
Yeah.
is screwed up too.
Yeah.
I mean, you have a great science establishment here. You're going to trash it, and then all the public health that comes from it is going to go down the tubes.
And it's going to take decades to rebuild this.
Yeah.
Because you can't just flip the switch off and then flip it back on and expect everything to work the way it did before.
Yeah, I think it's a lot easier to flip the switch off than it is to flip it on.
Really kills me? So, the first week he kills U.S. Agency for International Development, right? And then the court says, you can't do that. But it's too late. Everybody's fired.
Right.
And a funny thing, when you fire people, they go look for other jobs. And the ones who find other jobs fastest are the ones who were your best qualified employees, and you're not getting them back.
So
The ones you'll get back are the ones who couldn't find anything else.
it's just, there's nothing you can do.
No.
because
You
it's
can
all
do
shot.
TWiV.
Yes, you can do TWiV.
But I think
And for those who are going to protests this weekend, more power to you. Yep.
we made a big difference by helping people in other countries with that money. And I think it's unfair to just rip it out.
It's not just unfair, it's stupid. It's short-sighted, it's tactically ridiculous a blunder.
We have a paper for you, which
Get back to
Pretty in-
science.
pretty amazing, actually.
Yeah.
I like this.
Yeah.
This is really. How'd you come up with this one, Vincent?
I was just screening the tables of contents,
Hmm.
and I saw this. A natural experiment on the effect of herpes-zoster vaccination on dementia. And I said, well, I don't know anything about dementia, so let me see. And I said,
Yeah.
well, I don't have to know anything about
It's
dementia
a great paper.
to
It's,
understand
first,
what's going on here.
Co-first authors are Marcus Eiting and Min Ji. Senior author is Pascal Geldsetzer. And the crew is from Stanford University, the Leibniz Institute for Financial Research, SAFE, Frankfurt am Main, Johannes Gutenberg University, Mainz, Germany, Heidelberg University Hospital in Heidelberg, Vienna University of Economics and Business, and the Chan Zuckerberg Biohub in San Francisco.
You want to summarize it for us, Alan?
Yes, so this one I actually did write a little summary for. So there's some evidence linking dementia to Alzheimer's disease is what you think of, but in this paper we're going to talk about dementia as a general category for reasons that'll become plain later. So there's some evidence linking dementia to herpes virus infection or reactivation, but it's all been correlational, and that's been hard to test whether that link is real and whether we can use it to try to reduce dementia rates. This group took advantage of a natural experiment in the country of Wales, where the way they rolled out their shingles vaccine, now remember shingles is caused by varicella-zoster virus, which is a herpes virus, and in Wales what they did was people born after September 2nd, 1933 were eligible for the shingles vaccination. People born before that date weren't. I don't know exactly why that was done, but it was done. So this formed a natural experiment where you have people who were born 1 week who weren't eligible to get the vaccine, people born the next week who were, and if you looked at the whole population from those two, they didn't just look for 1 week, but if you looked at the before and after populations, you should be looking at an overall population that didn't have any other radical changes on that exact date. So you should have a good population for a control and an experimental population. So they looked at electronic health data. They found about 47% of the people born after the cutoff date had gotten the vaccination. Hardly anyone born before the cutoff date had broken the rules and gotten it. So it was like 0.01%. So this natural experiment was a go. So then they looked at a 7-year follow-up period, and they found that the vaccine-eligible group was not only much less likely to get shingles, because that's what the vaccine does, but also less likely to develop dementia. And then they confirmed this result in data from England, which rolled out the shingles vaccine in a similar way. And finally, they did a bunch more statistical analyses to look at possible explanations for this effect. Those implicate the vaccine's ability to reduce reactivation of varicella-zoster virus and also point to possible immunological effects of the vaccine. And the importance of this is it's the strongest evidence, I think, that we've got to date linking herpes virus reactivation to dementia, and it suggests a simple, cheap intervention that could reduce dementia rates by a significant margin.
I am willing to bet that the U.S. president has not had a shingles vaccine.
This, by the way, has funding from the National Institutes of Health.
Yes.
So this is how to vaccinate against dementia, if you like, if you want to
Yeah.
put it in a...
Yeah. Well, it's a side plus because if you're 50, you should get vaccinated against herpes zoster. So it's a side effect.
Alan was questioning the cutoff of that particular date, and that is a date for the people to whom that applied were 80.
Yes.
Okay, so it's as if we don't have an infinite amount of vaccine.
Right.
So if you're 80 or older, forget it.
And you haven't had shingles yet, or you already had shingles.
Yeah, you probably won't get it, or we don't really
probably
care.
the least of your worries. Okay, that's fair.
Yeah. So there's been evidence that dementia. Well, by the way, what is dementia? A general term for a group of brain disorders, including declines in cognitive abilities, memory, thinking, reasoning, and behavior. So cognitive abilities, not motor functions. That's other things, right? That's what dementia is. That's all you need to know for this paper. So, as Alan said, there's been some evidence that neurotropic herpes viruses are involved in dementia. And since we have a nice varicella-zoster vaccine, actually, this is done with the older.
Yeah, so this is Zostavax, not Shingrix. Which is
so
a point
Shingrix is a shingles vaccine, but we used to give the attenuated vaccine before, which is not as good as Shingrix. But I don't know if Shingrix would have the same effect.
That's right. I want to see. So
you'd have to redo it.
These two vaccines are as different as it gets, right? So the zoster vaccine is a live attenuated VZV virus that was initially developed as a chickenpox vaccine. Chickenpox and shingles are related, right? You get chickenpox, it goes dormant in your nerves, reemerges as shingles, same genome, okay? And that vaccine always creeped me out just a little bit because the vaccine itself can go latent, right?
Yeah.
And so, but it's proven to be, a perfectly reasonable vaccine. There haven't been serious side effects that have shown up in, God, it's been used for easily over 20 years, I think, maybe 30 years. But the Shingrix vaccine is a single protein from the surface of the virus. So it's a protein vaccine, no virus or anything else. So it's just one protein as opposed to something that's going to express the whole genome and go latent and probably express proteins for a long time. So mechanistically, it's quite different. And I don't know whether it'd be a very interesting question to see if both vaccines did the same thing in this.
If the mechanism is what this paper is implying, then the Shingrix should actually work better.
Yeah.
Because it is, it's better at preventing shingles, and that reactivation is what they're theorizing here. Is what's contributing to dementia.
Now,
but we'll see.
So before Shingrix, the zoster vaccine was just the chickenpox vaccine, right?
Right.
Yes, in given to adults in like a double dose.
Yeah. Okay.
By the way, we have had episodes on herpes virus in Alzheimer's. There's a twin here, twin 57.
Yeah.
This week in neuroscience from a month ago, and then there's TWiV 519 from eons ago, 2018, looking at a role of herpes simplex type 1 as a cause of Alzheimer's disease.
Now, the beauty of this study is that most studies say you want to know if a vaccine has an impact on dementia. You just take electronic health records and you compare the occurrence of dementia among those who got a vaccine and those who did not, right? But that's an observational study, which are always confounded with other variables. And you're assuming
confounded observational studies, yes.
You're assuming that all the things that differ between those who got vaccinated and those who did not have been completely measured and modeled and don't have anything to do. And they
all had equal access to health care. They all took equally good care of themselves. And
They say basically this is implausible. This assumption of no confounding bias is implausible because it's a lot of the data you can't even measure and you can't even verify it. So you can do an observational study with a diverse population, but it doesn't prove anything. It suggests always. And I don't care what people say, unless you can do a study like this, you're always going to have confounding variables, including ones you don't even know about. That's the point. So you can't even address them. And so this is different because of this interesting situation in Wales, because starting on September 1st, 2013, anyone born on or after 2nd September 1933 were eligible for zoster vaccination for a year, and then the older ones are never eligible, right, for reasons we just talked about. And so they can get these electronic data and compare adults who were ineligible for the vaccine, assuming they didn't cheat somehow, with those born after who were eligible. So basically, they're born a few weeks apart, right?
Yep.
And you don't expect any big differences. And you don't expect confounding variables, right? Because they haven't had time to develop diseases or lifestyles or whatever that would be a confounding variable. So that's brilliant. It's like the paper we did on the association of EBV infection with MS. You know, everybody has EBV. But it turns out that in the Army, when people come in, they take their blood, and a good fraction are EBV negative. So they could, and they take blood regularly from you, apparently, in the Army, and they could tell when you go EBV positive.
Through various means, yeah.
And then they can tell if you get MS, whether you're EBV positive or not. It was beautiful. Perfect study. Right?
There are so many of these papers that we do that are possible because of good records.
Yes.
Yeah.
You know, and if we had some of these come from companies, from countries that maintain a national health database, okay? And it's so useful and makes so much sense, you know.
Like this paper.
Yeah,
It's Wales,
exactly.
part of the UK. It has electronic health records for everybody. So you can mine the anonymized data and find stuff out.
So as I understand it, we're talking about really a population from just a 2-week window of time, right? A week before this cutoff and a week after. But that's I'm trying to dig out the numbers, but it's on the order of 250,000 people. So it's a large study.
Yeah.
Yep. Okay, so they actually looked at these individuals and show that they really don't have any major differences in, except for vaccine uptake, in other health services, common diseases, and so forth, educational attainment. All right. So the first thing they do is say, can we look at this data and conclude that vaccination prevents shingles? Right? Because that's a test of what you're trying to do.
Yeah, it's a control.
It's a nice control, right? And
so now remember, in these subsequent experiments, they're not separating out the ones who got the vaccine.
Mm-hmm.
They're doing this just by the date. So the experimental population is the group that's born after that date that could have gotten the vaccine, and it turns out almost half of them did. And then the ones before that couldn't get the vaccine. It turns out almost none of them did.
And it occurs to me that although they do in some analyses split out the people who got the vaccine, that potentially, it occurs to me, introduces another variable, because people who are inclined to take the vaccine may be a subset of the population who are more inclined to take care of themselves. Okay?
That is still possible.
Still possible.
Yeah, I was just looking to see if they addressed that in some way, but I don't know that you could. So clinical trials have shown that Zostavax reduces the occurrence of shingles. Okay, so they look at their data and they look at the occurrence of shingles in adults born on either side of the birth date, right, for eligibility. Right, that's really cool, and it's not whether someone got a shingles diagnosis. It wasn't whether they got the vaccine, but whether they got a shingles diagnosis or not. So they have a follow-up period of 7 years, all through records. 14,465 among 296,000 adults had at least one diagnosis of shingles. And being eligible for the vaccine reduced the probability of having one shingles diagnosis by 20%, relative reduction of 20%. Okay. And if they add the data on whether they got the vaccine or not, it goes to 37%, which is cool, right? And that's about what they saw in the clinical trials of Zostavax. So there's a beautiful control here, right?
Yeah.
Okay. Now, what about dementia? So they define dementia as any kind of dementia, like the things I told you just before. That's the outcome. And basically, if there's a diagnosis of dementia on the electronic health record, or if the cause of death on the death certificate was listed as the primary or contributing, was dementia listed as the primary or contributing cause of death, right? And so during this 7-year follow-up, 35,000 of these 280-some thousand people were newly diagnosed with dementia. There's a lot of dementia. It's like 10%. It's really not good, right?
Nope, it's out there.
Yep.
Being human is a problem.
Yeah. Aging. That's the issue.
Yeah.
Evolution expected us to be dead by 40.
Absolutely.
That's right. That's right. Yeah.
Just get your children to reproductive age, and then
That's right. And you're done.
You're done.
You're
so the effect of being eligible for a zoster vaccine, remember, that's the one way they looked at it, is relative reduction of 8.5% in the probability of a new dementia diagnosis over those 7 years. So just whether you were able to get the vaccine, but actually, if you actually got the vaccine, the reduction relative reduction of new dementia is 20%. That's a lot.
Right. So to Rich's point about how the people who got the vaccine may just take better care of themselves, same problem all the observational studies had, that same phenomenon of some people taking better care of themselves would exist at equal rates in the people who couldn't get the vaccine.
Ah, true. Okay.
Therefore,
yes.
This difference...
Fine.
How do you know that, though?
Because they were born a week apart. I mean, why would 150,000 people who were born the previous week versus 150,000 born the next week have more or fewer people in their population that take good care of themselves? They wouldn't.
Yeah. You're right. That's the point of this 2-week period
That's the
with
point
an
of this.
arbitrary cutoff.
So then, you've got an equal number of, there's some population within any group that are going to be more diligent.
Yeah.
But if you were diligent and you missed the cutoff for this, fine, you were diligent.
You didn't have a choice.
You didn't have a choice, but you're the control group in this natural experiment. And now it turns out that the vaccine is the eligibility for the vaccine is the one difference between these two groups.
Yeah, so the listeners can delete my previous comment.
That has this significant protective effect.
Yep.
But the next analysis, I think, addresses this also, right? So they say we can look at other things that abruptly change on 2nd September 1933, right? The cutoff date of birth. So they looked at 10 other leading causes of disability, adjusted life years and mortality, and no effect of zoster vaccination on any of them. So the idea that it's just people that want to be vaccinated is not supported by that either, right? Okay.
And also any idea that something happened September 2nd that suddenly made people, a lot healthier that had nothing to do with the vaccine. That's not the case either.
Aliens, man.
Aliens landed on September 2nd, right?
They also, they're very concerned about this date, something happening at this date. So they did 4 other things to make sure they weren't being fooled. First, they show, so this is actually interesting, that this September 33 date threshold does not affect the probability of taking up other preventive health interventions, which is just what we were talking about,
Yep.
right? Secondly, they examine whether this date is also used by other interventions that affect dementia risk, and it doesn't. Third, they find no difference in the 7-year incidence of dementia between age cohorts around 2nd September 33 for the 7-year period before the zoster vaccine rollout. And fourth, using 2011 census data, there are no discontinuities across this date in the proportion of individuals in Wales who reached a particular level of education, right? So that's interesting, because you could argue that that might lead to dementia
Right.
as well.
Right. If there was some educational policy change, or suddenly everybody started going to university and
Or they had to memorize the Krebs cycle and it drove people
Oh,
crazy.
my God.
Okay. All right. So this is all now about showing that their analysis is robust. You already know the result.
Yeah.
And they want to convince you because this is all
Because it's a Nature
statistical.
paper.
It's a Nature
And
paper.
this is
And also,
important.
this is statistics, right?
Yeah.
So they have to do this. They have two different ways. So they've been using what's called a discontinuity. What's the name of it? Regression discontinuity approach, right?
Yeah.
Around this date. And they have a different way of doing this, looking at the same data. It's called difference in differences instrumental variable analysis, which takes advantage of the fact that this September 2nd birth date is well, that's what they're using here as the cutoff for the zoster vaccination program. And so basically, the two regression approaches, DID4 and regression discontinuity, give you the same result, a good reduction in the case of dementia, and also shingles and post-herpetic neuralgia, which is another condition that can be helped by the shingles vaccine. All right, so that's good. And all right, so this all looks good, right? The statistics make sense. What's the mechanism? How is immunizing with Zostavax helping prevent dementia? So they have three ideas. First, changes in healthcare pathways as a result of a shingles episode. So
somebody gets shingles, they go to the doctor, they end up on some medication, it's the healthcare pathway they take is different from the pathway of somebody who doesn't get shingles or gets a very mild case.
Two, a reduction in reactivations of the virus, because that's what the vaccine is, I suppose, supposed to be doing. Although I don't know if it's a reduction in reactivation or the virus reactivates, but then the antibodies from your vaccine are neutralizing it, right? I don't know that the vaccine prevents reactivation. Do you, Rich?
No, I don't know.
Yeah. Anyway, so that's something to do with the virus.
Reactivation is a very squishy topic, as
Yes.
you've already implied, because you count it as a reactivation when you can detect it, right?
Yeah.
But
Yeah.
there's probably, in my mind at least, a whole lot of activity going on that flies under the radar. So it's a matter of whether it reaches a level of detection and what your detection mechanisms are. And, as you already implied, there could be a whole bunch of antigens being made, other stuff going on under the radar.
All right. And the third thing is some kind of immunomodulatory effect induced by the vaccine, right? Independent of the virus, antibodies or interferons or something that's causing the suppression of dementia, right? So they try and work through each of these. So changes in healthcare, what they say is reduced healthcare use resulting from a prevention of shingles, right? If you get vaccinated, you don't have shingles, so you have reduced healthcare interaction, fewer opportunities for you to be diagnosed with dementia, or implementing care changes that would increase the risk of being diagnosed, right? So they say that their sample size, the effect of the reductions in shingles is too small to be accounting for this. But anyway, they did 5 different analyses to examine this healthcare change idea in some detail. So the first is that if shingles presented an opportunity for the health system to diagnose dementia, they should also diagnose other chronic conditions, right? So they applied the same regression analysis that they have done already with dementia to the top 10 leading causes of dementia, right? In this group in Wales, and with the exception of rheumatological diseases, the zoster vaccine has no effect on any of the other chronic diseases. They don't talk about rheumatological diseases anymore. And second, they adjust their regressions for the frequency of health service use, no effect on that. Third, they implement, they did their analysis on people who visit the provider at least once a year during each of the 5 years before the start of the rollout, and the effects are the same. And fourth, they added whether individuals had a shingles episode during the follow-up as a covariate in their primary regression discontinuity analysis, which is the statistical approach that they use. And they found that adjusting their analysis for shingles episodes did not change the point estimate. And finally, fifth, they did a study among participants that got a shingles diagnosis to investigate whether episodes of shingles led to changes in health care received by patients. And they find that shingles diagnoses did not increase the probability of getting a new dementia diagnosis in the months after. So it seems to rule all that out. Changes in health care don't explain their findings. Are we good with that?
Yeah.
I'm good with that.
I buy it. All right, what about reduction in reactivation of VZV? So they look at the time during the follow-up period when the effect of zoster vaccination on dementia appears to begin. And so they plot incidence curves for dementia for those who are eligible versus ineligible for zoster. So if the mechanism is through reduction in VZV reactivation, you would expect that the effects of the vaccine would begin before observing an effect on dementia, right? They should have an effect on but the reduction in the incidence of dementia begins only after more than a year. So the VZV effect precedes it. So not necessarily consistent with VZV reactivation being part of it. Second, they do propensity score matching to compare the association with dementia from experience.
I do want to point out that they did find antiviral treatment for a shingles episode is associated with reduced incidence of dementia.
Did I miss
That's
that?
a supplementary.
Yes.
So it's actually the data on this are a little mixed. There may be something to do with viral reactivation, but not clear.
Yeah, they say if the mechanism is through reduction in reactivations, you would expect to see an effect on reactivation before you see an effect on dementia, and that's what they find. Right.
And they also found people that had multiple rounds of shingles, multiple reactivations, had a higher risk of dementia.
Yes, that's right. Yeah, that's the next one. Second thing, higher incidence of dementia if you have more shingles episodes. And third is the antiviral medication. Antiviral treatment of a shingles is associated with a reduced incidence of dementia. So I don't know, maybe the timing thing is just not accurate, right?
Maybe.
Because the vaccine works in a couple of weeks, and maybe dementia just takes longer, right? But still is linked to virus, which these other points actually are consistent with. All right. Finally, they look at this idea that there's some immunomodulatory effect, which is independent of the virus. And one of the things that is useful here is that pathogen-independent immunomodulatory effects after vaccination tend to vary strongly by sex and depend on some other vaccines either before or at the same time. Okay, so their effect of zoster on dementia was much greater among women than men. I didn't mention that, but that's what the data say. So that's consistent with some kind of immunomodulatory effect. They also find, this is interesting, that their protective effect of zoster on dementia is greater among people who did not recently get the influenza vaccine, which they're saying is consistent with an immunomodulatory effect. They're not actually proving it. They have to do that. Well, they're saying it's consistent with that. And finally, they look at the effect of zoster vaccine on dementia, comparing people with or without an autoimmune allergic condition. Okay, the idea here is that the incidence of shingles is increased among people with an autoimmune or allergic condition, right? And so these are characterized by heightened activation of the adaptive system. So they find that the zoster vaccine has a stronger effect on preventing dementia among those without an autoimmune or allergic condition. So they feel in the end that there are two things going on here. You're interfering with VZV reactivation, and there's some kind of immunomodulatory effect independent of the virus. And obviously, you're going to have to do more studies to show that. But 1/5 reduction in dementia by this Zostavax.
That's better than these trumpeted drugs that are, trying to target Alzheimer's disease, and you get a side benefit of not getting shingles, and, you don't get the side effects of these Alzheimer's drugs.
So most of the research, as I understand it, that links herpes virus infections with dementia, the linkage is with HSV1, right?
Yeah, that's right.
Yeah.
Though they point out here in the discussion that VZV does things that seem like they could contribute to dementia, though, as I understand it, the literature doesn't make as direct a connection as with HSV1. And they also, in their discussion, talk about how a VZV reactivation in the brain could cause, how do they describe it here? But a neuroinflammatory response that would have an impact on that could have an impact on HSV1 as well. So that if HSV1 is the culprit, VZV reactivation or whatever could impact indirectly through immune mechanisms on HSV1. So clearly we need to know more about other herpes viruses, neurotropic herpes viruses like VZV on dementia, and I'm sure that's coming down the pike. And it also makes me think, making herpes virus vaccines has been difficult. The only one I know of is the varicella-zoster vaccines, right? And yet these are quite similar, viruses. I don't understand why there has been difficulty in developing, or maybe it has to do with the amount of effort that goes into it, and how dangerous the different viruses are that motivates the research. But why don't we have an HSV1 vaccine?
Mm-hmm.
I think the funding issue is big there.
Right. So chickenpox.
I mean, why? What's what's your grant proposal going to look like for vaccinating against HSV1? We're going to stop cold sores.
Right.
Yeah, okay.
On the other hand, if your grant proposal now is we're going to stop dementia,
yeah,
that puts a different spin on it.
Gives you a much better selling point.
So they talk about some ways that VZV has been linked to dementia. So VZV reactivations have been found to cause cognitive impairment through vasculopathy, amyloid deposition, aggregation of tau proteins, neuroinflammation, and a variety of cerebrovascular diseases, ischemia, infarction, and hemorrhage. So a lot of things happen after VZV infection. Now, the thing is, this is Zostavax, which is no longer used, right, as a shingles vaccine, because Shingrix is much better, actually. It's almost 100% preventing shingles. So I don't know what to do about this. So the people who've already got Zostavax, great. They're going to be somewhat protected, but what do you do about the rest of the people? Do you find the mechanism and find another way to attack it rather than Shingrix or Zostavax?
I think you continue with Shingrix because the evidence here is suggestive that there is a viral role and that stopping reactivation or slowing reactivation is going to be useful. And the immunomodulatory effect, we don't know exactly what the mechanism is there. But if it's antibody-based and we're eliciting an antibody response against the same virus, we're in the same ballpark.
Yeah.
So I think you continue with Shingrix and hopefully find some correlative data that you're going to get a similar effect.
Yeah, you can't do this kind of study with Shingrix, obviously.
We didn't have the same. I mean, unless there's some country that rolled out Shingrix in this same conveniently timed way.
Well, there certainly are other countries that at least have large countrywide electronic medical databases from which some data can be extracted.
Well, and in fact, the changeover from Zostavax to Shingrix occurred in a fairly short time period.
Yeah.
Most places, I suspect. So you could look at that. You could do before and after analysis of that and see if you see changes because if Shingrix is useless against dementia, then you should see the reverse of the effect you see in this paper. If Shingrix is equally useful, you should see steady line.
So the switch to Shingrix was in September 2023.
Right.
So you could look at records right around that date and see incidents of dementia, right?
Yeah.
Interesting. Okay, well, I got Shingrix, two doses.
I got both. I got
Shingrix.
Zostavax and Shingrix.
You did?
Yeah.
Yeah.
And I'm feeling pretty good.
I didn't feel so good after the second dose of Shingrix.
Well,
that was a rough ride.
You know, Rich is on TWiV. He's on his feet, so we can monitor him.
Yeah, just keep your eyes on me, and you'll have the answer.
When you say that herpes is an RNA virus, then we'll start the question. Okay. Let's do a couple of emails. Rich, can you take the first one?
Right. David writes, "Hi, TWiVniks. With regard to diarrhea virus diseases, I may have missed this point on TWiV 1199 or in an earlier epitope. So if I have, forgive me. If the purpose of the host immune response induced diarrhea is to flush the virus quickly to protect the host, maybe the purpose of the second phase viral protein induced diarrhea is to flush the newly replicated virus into the environment to prevent it from getting trapped in the GI. Just a thought. Love your work, and Miss Dixon. Best regards, Dave." And this is David Spector, from emeritus professor from Penn State, who writes us frequently. So
that's a good idea.
I'm kind of blanking on this.
So remember that paper we did. There's an innate response to norovirus.
Right.
And that causes diarrhea.
Right.
But then there's a viral protein made that also causes diarrhea, which presumably is made a bit later. And so he's suggesting that maybe that by then you've got virus particles made, and that helps flush them out.
Okay.
Yep.
Sure.
I like it. It's a good idea. I like it. All right, Alan, can you take Michael's, please?
Sure. Michael writes, Dear Twivers, my condolences on the loss of Dr. Dixon Despommier. I am an epidemiologist, and the majority of work in the past 2 decades has been on Alzheimer's disease, which is about as far away as one may get from the viral level. See today's episode. I started listening to TWiV during the beginning of the COVID pandemic and still listen to TWiV, and I also appreciate Dr. Griffin's clinical update. One of the concerns raised with the new leaders of the U.S. Department of Health and Human Services regarding a vaccine for poultry is viral reassortment. I was wondering if you might address this topic. RFK Jr., as head of the U.S. Department of Health and Human Services, made some comments that were reported by CBS News. CBS News refers to an interview on Fox News with RFK Jr., but I couldn't find it. I want to pull out some of his quotes from the CBS News website. And pulls out these quotes. So quote number 1, there's no this is RFK Jr. speaking, there's no indication that those vaccines actually provide sterilizing immunity, and all 3 of my health agencies, NIH, CDC, and FDA, the acting heads of those agencies have all recommended against the use of the bird flu vaccine. And this is now Michael's question. I am not aware of any vaccine that provides sterilizing immunity. Is this correct? I don't think there's a vaccine that provides 100% sterilizing immunity.
That's right. There'd be no reason to expect it in chickens.
Yeah. Quote number 2 from RFK Kennedy said, without sterilizing immunity, it would amount to, quote, turning those birds into mutant factories, resulting in worrying genetic changes to the virus. Quote, that could actually accelerate the jump to human beings. Question, wouldn't this be a concern of any vaccination in any population, including humans? Yeah, and in fact, they're more likely to be mutant factories if they are hosting active infections, which you are less likely to get if you vaccinated them.
Yeah.
So
we don't see. I mean, there's some pressure exerted on influenza virus by the flu vaccines, probably still select for antigenic variants, but for the other viruses, we do not see that vaccines are putting pressure to select viruses that are whatever more, right? No.
As usual, RFK Jr. is 180 degrees wrong. Quote number 3, we've in fact said at the USDA that they should consider maybe the possibility of letting it run through the flock so that we can identify the birds and preserve the birds that are immune to it. Question and comment. Comment, the CBS News site reports that USDA had a different opinion under Biden. Yes. Question, wouldn't this, quote, letting it run through the flock be as bad as the concern raised about the lack of sterilizing immunity in the vaccines? Yes.
Yeah.
I am not aware of naturally acquired immunity providing sterilizing immunity. Do you know of examples and how common this might be? Again, I don't know of examples of natural immunity being sterilizing. And I will also point out that RFK Jr. not only doesn't understand vaccines, he doesn't understand chicken farming.
No, his 2 scenarios are he wants he's concerned about vaccines being mutant factories causing mutant but he wants to let the virus rip through,
and the way chicken farming works is identify the birds and preserve the birds that are immune to it. Well, guess what? The birds that are immune to it are either going to be broilers or at which you're not going to be able to breed because, after you broil them, they just they don't do a whole lot. Or they might be laying hens, in which case you could get some eggs from them, but this is really not going to work to regenerate a flock, and immunity is not exactly inherited, so this doesn't work on any level.
It's also the genetic diversity of these chickens are very restricted, so it's not clear that
it's
would
not
be
clear
variants.
You're going to get any that are going to be useful variants that'll be, super immune chicken.
Isn't it interesting how, so the USDA, the people are basically the same, only the director is different, and now they have a different approach.
Yeah, it's weird.
And the director is a lawyer and a politician who knows no science.
Yep.
I question the whole idea that letting it run through the flock would generate any resistant birds. I mean, that's just kind of
there's no guarantee that would happen.
No.
And there's no guarantee it would be an inheritable trait that you could pass on to any future birds.
Boy, gosh.
Thank you,
All
Mike.
right. Matthew writes, I was wondering how measles could spread to, let us say, West Texas. I came up with this hypothetical question that I cannot find an answer to. I am immune to measles. I travel to an area of the planet where measles is spreading and infecting unvaccinated people. Can I acquire the virus, return to the U.S., and spread it, even though the virus cannot replicate in any of my cells? Okay, Matthew, what happens is you shed virus for some days before you get a rash.
So the statement, it can't replicate in any of your cells, that's not how immunity works. It can replicate in your cells, but your immune system generates antibodies against it.
But also, vaccinated people are not known to transmit very efficiently. So you would have a person who was unvaccinated in another country, picks up measles or somewhere else in the U.S., is in the incubation period and goes to Texas.
Yep.
It's quite straightforward.
It's unvaccinated people is the issue.
Yes.
And there's enough of that sort of stuff going around so that there's always measles out there.
Yeah.
But it doesn't pop up unless the vaccination rate in a given population is low enough so you get spread.
And unvaccinated is not necessarily idiots. They might be too young to have had the vaccine or to have had the full series.
Yep.
So somebody is going to visit relatives in West Texas, and they just had a baby, and so they're taking the baby to see the relatives, and they take the baby on the plane, and the baby has measles. There you go.
Rich, can you take the next one?
Sure. Brian writes, Dear TWiVers, although there is much that is merely chaotic and destructive with Musk's gutting programs, the ending of the president's malaria initiative is probably deliberate. At least 120,000 Africans, mostly children, will die in the next 12 months from malaria. A leaked USAID memo puts the death toll at above 160,000. These children would survive, except for Mr. Musk's actions. I find it important to explain that malaria is a very difficult vaccine target. It is a protozoan. SARS has 30,000 base pairs, malaria 25 million. When injected via mosquito bite, the Plasmodium falciparum sporozoite homes in the liver, reaching it in as little as an hour. The only widely tested vaccines target the sporozoite surface proteins. They require continuously repeated injections for antibody production. Memory B cells simply do not have time to differentiate into antibody-secreting plasmablasts in the brief window of time before the sporozoite enters the liver, sheds surface proteins, and divides. But you can stop a malaria infection in its tracks with inexpensive medicines. You can prevent malaria infections with bed nets and a compound mosquitoes avoid, and discrete applications of insecticides to the walls where mosquitoes live. In the 2005 George W. Bush administration established the President's Malaria Initiative, its goal to reduce malaria mortality by 50% in Africa in 10 years. The initiative, run by USAID, exceeded its goal. In a decade, malaria mortality was reduced over 60% in Africa. The program provided bed nets, insecticides, anti-malarial drugs to treat children and pregnant women who had gotten the disease. They were the most likely ones to die. Perhaps 800,000 Africans died annually from malaria in the early 2000s. By 2019, that had fallen to less than 300,000. 500,000 people, mostly children, saved every year because of the USAID program at a cost of about $500 million a year. Getting local people into rural areas with carefully applied resources, the key thing is that African people are eager for solutions. I did some research, ran a very small development project, started a cooperative in a couple of West African countries. I talked to women who had lost all or most of their children, most to malaria. They were the saddest people I've ever met. People in areas where malaria is endemic want solutions. The president's malaria initiative cut malaria deaths so dramatically. Malaria deaths so dramatically has been stopped, ended by this administration. But here I take issue with the idea that this is a random act of cruelty. This is not President Trump's idea, it's Musk's. He's the juggernaut attacking USAID. Mr. Musk is South African. He has had malaria. He knows what USAID does, as do most Africans. He knows thousands of African children will die each month going forward. Apply Occam's razor. Martin Luther King said South African apartheid was the world's worst racism. It is the simplest explanation for Mr. Musk's actions. I live in the San Francisco Bay Area, where Musk still has a cult following. I object to this conclusion. They don't understand apartheid. The estimates of predicted deaths are robust. During COVID, anti-malarial services were reduced some, and malarial deaths increased by 80,000. Those services are ended. 120,000 extra deaths is conservative. Thanks for all you do, Brian.
God, I hope you're not right.
I hope you're not right.
That is dark and grim, and I can't say you're wrong.
Okay, next one, Alan, Sergio.
Yeah, so Sergio writes, "Hello, TWiVers. I was so sad to read about Dixon's passing. I am an agronomist interested in plant pathogens, and that is how I started following TWiV when I was doing my master's in Japan. Then I was amazed about his interest in vertical farming and bought his book on the matter when I did my postdoc in Sweden. Now I am back to Bolivia lecturing microbiology to biotech students. I just wanted you and everybody who knew him to know how big was his impact on my career. I hope my short message helps all of you overcome the pain of losing such a charming and interesting person. My condolences to all of you." And Sergio has a PhD in biocontrol science and is at Tokyo University of Agriculture and Technology. Thank you, Sergio.
Ryan writes, "Please be advised, Trump will directly overturn vaccine laws, all to hold on to RFK Jr. supporters." He sends a USA Today story about Trump signing an executive order prohibiting federal funds from going to K-12 schools and university that require COVID vaccinations. In Iowa, that shouldn't be a problem. The state has already outlawed mandatory vaccinations. Trump's order instructs the Departments of Education and Health and Human Services to issue guidelines to comply with the order and create plans to end coercive COVID-19 vaccine mandates. That solves the problems, he said after signing the order. As in Iowa, the order will likely have a minimal impact. No states currently require COVID vaccinations for students at K-12 public schools, and at least 21 states have passed laws banning vaccine mandates.
This is just throwing red meat to his supporters. It has nothing to do
politics,
with anything.
right?
Yeah.
Just politics. And Richard, let me take the last one really short. Richard sends an article in Ars Technica. It appears all we can do is watch, continue to point out the problem, and circle the wagons. Thank you for continuing to supply the best scientific information on infectious diseases. And this is an article that we talked about yesterday on Clinical Update. It's in Ars Technica. CDC buried a measles forecast that stressed the need for vaccinations. They're about to release a report on that, and the powers that be cut it off. They'd be saying that the decision to vaccinate is a personal one, which it's not. And they said, people know this anyway. There's no need for us to release this information. The worst possible excuse for withdrawing it. It's just not consistent with RFK Jr.'s beliefs about vaccines. So this is the kind of world we're in, folks.
Yep.
Time for some picks of the week. Rich, what do you have for us?
Another one of my hot science picks. This one is from the American Osteopathic Association. I forget where I got this. And it's titled, "Researchers Find Certain Roller Coasters May Help Small Kidney Stones Pass." So apparently there has been anecdotal evidence of people riding roller coasters and passing kidney stones as a result. I don't know if that's during the ride or afterwards, whatever. Okay. And actually, the roller coaster, it says certain roller coasters. The one that really pops up as a winner is the Thunder Mountain roller coaster in Disneyland or Disney World. I think they have one.
Disney World.
In Disney World.
I think so.
Okay, at any rate. Which, is not a real extreme roller coaster. It's, from my point of view, it's a pretty mild roller coaster. At any rate, there was anecdotal evidence for this, so some clever individuals decided to do the experiment. And they have these model kidneys. Okay, I don't know. They're fluid-filled. They're somehow model
3D
kidney.
printed a model kidney. It was a model of a specific patient's kidney.
Right. And they took these model kidneys and loaded them either with small, medium, or large things that would mimic kidney stones, and then took each of them on 20 different roller coaster rides to see if the model kidney, they stuffed them in their backpacks, right, and rolled the roller coaster. To see
They got permission from Disney to do this.
To see if the roller coaster would pass the kidney stones, and sure enough, they did. Okay, so there's support for that hypothesis. I thought you might want to know that.
Thunder Mountain is nothing. Oh my
I
gosh.
know.
Yeah.
Even I go on that.
Yeah.
And they found, which will not be surprising to people who've ridden roller coasters, the back car is the best.
I thought that was
significantly better than the front car.
Yep.
Interesting.
Absolutely.
So Rich, what do you think is a tough roller coaster?
Oh, I don't know. I mean, it's been a while since I've been on a roller coaster, and I'm sure the technology has developed significantly. But there's this thing called Zumba in Busch Gardens in Florida that's pretty good. Okay, and there were a couple of others in this place in Niagara Falls, but I'm far from an expert. It's got to go upside down, man. Okay, or it's just not worth your time. There's also this really weird device. I forget where this was. This may have been Busch Gardens as well, where you sit in a chair on the end of an arm, and it just goes all over the place and shakes you around. Okay. I think that might be worthwhile looking into.
I think I went to Busch. They have an amazing collection of roller coasters. Yeah. But the one that they just knocked one down at Six Flags called King the Ka. It was a hairpin. The cars went straight up, and then they just went straight down.
Oh.
Right?
Yeah,
and,
cool.
I mean, you don't go upside down, but that
ought to do it.
On.
I would think. Coming out of the bottom of that ought to do it.
Scariest coaster I ever rode was the Cyclone at Coney Island. Not because it's an extreme coaster in the usual sense, but because I don't even know if it's still there. I think they may have torn it down, but as you were going up the hill of this like 80-year-old wooden coaster that's out there in the salt air on Coney Island, New York, you could see the amount of dry rot and decay in the boards of this thing before it drops you off the end. And I'm thinking, gosh, I hope we don't make the news.
Yeah, that might actually make me pass a kidney stone before I even,
even if I didn't have one.
On it.
Yeah.
So I looked up the most extreme roller coaster, and King the Ka came up as number one. It was 456 feet tall, and the speed was 128 miles per hour.
Oh.
So the cars, I think there were 4 cars, they would go over the top and stop facing down, for some seconds.
Just to give you something to think about.
Yeah, looking straight down, then it would release. But there are others. There's another one at Six Flags, Intimidator, and then some other that I don't know about. But I don't like extreme roller coasters.
I've outgrown roller coasters.
But I just like to watch other people ride them, and they just love it. Some people just love it. It's amazing, right? Not for me. Alan, what do you have for us?
When you're feeling, when you get a kidney stone, you might want to reconsider.
Yes, if I from what I've heard of kidney stones, I would ride any coaster probably to get rid of one of those.
I don't know. That would be a toss-up for me. You can have them vibrated and destroyed,
There's
right?
probably, the clinical application of this would be to come up with some non-roller coaster based method that's less specific.
Alan, what do you have for us?
So this is a double pick. About 6 months ago, I discovered that these two magazines from my childhood still exist, and it just warmed my heart. And they have digital subscriptions. So I've linked to both. They're run by the same company now: Analog Science Fiction and Fact and Asimov's Science Fiction. And these are, if you've ever seen the paper editions, I got these occasionally as a kid when I would, buy one on a newsstand. They are printed on newsprint, and they're just cover to cover science fiction short stories.
Oh, okay.
I think Analog actually has some nonfiction. Actually, yeah, Analog has some nonfiction stuff in the front. But they're really the meat of the book is just, they'll have like 12 short stories in an issue. These come out there every other month, I think, or every few months. And you can subscribe digitally, and then you just, download an issue, and I've got it on my Kindle, and I can pick it up anytime and read a sci-fi short story. Good stuff.
Oh, that sounds great.
Yeah.
Let's go back a ways. These
go back, I think Analog is 90 years old or something, and Asimov's, was started by Asimov when he was an up-and-coming writer, and yeah.
It's cool. All right, I have an article in eLife by Ran Bleckman, How Science Under Threat in the U.S., How Scientists and Institutions Should Respond. And I want scientists to read this because he has a lot of good ideas of what people should do. He says, individual scientists cannot afford to retreat to the lab and hope this passes. We have to take action.
Especially since your lab probably lost funding.
Yeah. Lots of good things to do. But also buried in here, he says, you have to write something, but don't write it in a scientific journal so your colleagues read it. That's not the point. You're not trying to reach them. Write it somewhere else where people, regular non-scientists, are reading. And I see so many people publishing in Nature and Science and Journal of Virology why science is great. We already know it's great. You think Mom and Pop are reading Journal of Virology? No. MBio? No, they're not. So put it somewhere else. So this is good. Good points here for you.
Yeah.
Right into your local newspaper.
Yeah.
And write to your representatives and senators in Congress. I mean, unless you live in a state where your representatives and senators are right at the front lines of this already, in which case you can, just send them a hurrah occasionally, which is my case. But yeah, even if you're in a deep red state and you think, they're not going to listen to me, they listen to constituents because they know that their job depends on it, and that they will get from their staff a summary. We got this many people writing saying that they're mad about these cuts, and that matters.
Yep. My pick is, I already did my pick.
You did your pick.
Charles is a listener pick. Here's a pick I think people should read. Not that it will be enjoyable. It's in Tech Dirt. Why Tech Dirt is now a democracy blog, whether we like it or not, by Mike Masnick. And, he goes through the political scene, what's happening today, and criticizes a bunch of people. It's not fun because he's covering every bad thing that's happening here. And he says the future of American innovation isn't just another story we cover. It's the story, and we're going to keep telling it whether the powers that be like it or not.
He says here in his letter, it's a similar perspective, the article. So, similar perspective as Microbe TV and other medical research sites have. And I think, to me, what this is, as we've said before, this is not a political podcast, but when the politics impact the science that we are trying to deliver, then it becomes, we have to take that into account. And several other disciplines are experiencing the same thing.
Yeah.
He says, here's the bottom line. When WAPO's opinion pages are being gutted and tech CEOs are seeking pre-approval from authoritarians. The line between tech coverage and saving democracy has basically disappeared. It's all the same thing.
Yeah, that's it.
Yeah, we're going to do this work because someone has to. That's what I feel about Microbe TV. We're going to do it because we can, and we like to do it. That's TWiV 1207. You can find the show notes at microbe.tv/TWiV. We'd love to get your questions and comments and picks. You can send them to twiv@microbe.tv. And as I said earlier, if you like these programs, we'd love to have your financial support. Go to microbe.tv/contribute. Rich Conda is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at Microbe TV. We have a presence on Blue Sky. Microbe TV does. microbetv.bsky.social. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We will be back next week. Another TWiV is viral.